Thank you so much. That was great. Our idea of it would be, since we're going to have a panel discussion, that we would move on to the next talk and then break and then come back together for the panel. So unless somebody has a burning matter, I'd like to introduce Mei He. She is an assistant professor from Kansas State University. She is actually developing sensors for a number of uses, including cancer and environment and other biomedical applications. She is also, will learn more about some of her technologies tomorrow, since she also graciously agreed to sort of donate a scenario or two for tomorrow's scrimmage. So thank you very much, Meg.
All right. So I feel really grateful to be invited for this workshop, and thank you for hosting this workshop. It would be very interesting to invite people from different sectors to share the experience and the perspectives. And actually today I speak for the academia side and try to see what's the gaps and the challenges we can bring from the laboratory technology to the commercial products. So I want to share some experience and also the lessons and learn for the past commercialization road path. So I want to start first with this commercialization roadmap. This is really the picture I adapted from the review paper published on the 2015 from the Lab on Chip. I think it's great to summarize all the necessary steps and the factors that affect the technology development to the production into the markets. And as you can see, there are two parallel avenues here, and the one is for the customer development, the other one for the product development. And so speak from the academia side, so mostly we really focus on the product development, and we hope this is a great technology, and we test it and validate in the lab, and definitely it's going to be the great in the market. But the truth is not really in this case, and so that's really the two parallel avenues, but they need very frequent communication in between, and there are backfalls and between those two avenues, and to talk about to revise your products and finally put into the production and to put into the market. So definitely it needs a common language here for understanding this production road path from the academia perspectives, and also
in the back they're having a really hard time hearing you, so I don't know if you should
probably use the mic. Oh, okay. Okay. So here you can hear. Okay. Is it better? Oh, okay. I can use a handphone. Yeah, a little bit better. Okay. So hear better now. All right, great. So from this perspectives, and I say we really need to find a common language in between here to facilitate the customer development, and meanwhile with the production development from different sectors. And so here, this is really the first experience and also the lessons that I have learned before. So I want to share with you. It's not really a successful experience, but definitely the lessons and I learned. So that's the time I was a postdoc at UC Berkeley, and think this is a great campus. They provide very great facility for the technology transfer, and they provide free incubator space and the consulting program, and to help the students try to have the innovative technology translated into the market. So I was very excited at that time. And so the first, actually, the technology and development is really the photo-patentable nanomaterial. So this nanomaterial can be photo-patented in the regional location and change the pore size, the structure, and meanwhile you can change the chemistry of this nanomaterial and the patent in the different locations. So we come out with several patents. And this is the one which can, so this one actually which can patent in the 3 regions and with different pore size in the nanoscale, and you can immobilize antibodies on the surface of these nanomaterials, and try to automate and streamline the sample processing and the sensing steps in the one integrated chip system. And later, we think they can be used for the biomarker discovery because it's automation, and that's great to replace the old-fashioned laboratory technology. And So after the patent came out, and we work with the BioRed, it's a great biotechnology company as well, and then they licensed all the patents, and we're quite excited. We have the great industry partner, and we have the funding support this work as well. But finally, unfortunately, it did not reach the markets. And so that's really the lesson, and we try to figure out why it didn't get to the market and it didn't get a great sale. And so speaking of the purpose of the use, we try to focus on the streamline, the automation of the Western blotting. Probably most of you know this is a basic life-size tool in a conventional laboratory. It has to go through multiple steps, and it's labor-intensive and time-intensive, and then we hope we can use the nanomaterial and integrate with microfluidic technology to form one automated Western body, so which can substantially increase the efficiency and also the duration. Try to have that much faster to perform in the 2 minutes compared to the 12 hours used to be done in the laboratory lab for the biomarker discovery or the biomarker detection. So, but the major problem now is really what market you try to identify. So that's really the lessons we learned previously. The one size doesn't fit all. We have to specify what market, what exactly market we want to fit into. So later on, I landed my faculty position, but there are two graduate students, they continue to work on this startup company, and they spin off with university. Actually, most of the time, they focus on the other factors, like the strategy, the business model, and the marketing and the sale, and that integrate with the product development. It doesn't mean the good technology can be a good product. And then we have to develop this common language to understand what's the needs from the market and try to come back to revise the product. And this is really the back forth for multiple times. And actually, they spend a lot of efforts in this part to start a customer survey and to revise the products in the back forth. And finally, the efforts falls on the several factors here. It's going to be a reagent kits. It's for the clinical test, the research tools, and what market we can identify to move this technology to. And so this is a lot of efforts here, and finally they decide to focus on the research tool. And then later on, actually early of this year, and this technology has been put on the market, and the start at being accurate by the protein samples early of the year. But you can see the product here. It's totally different version than the original one we developed. So that really tells you how we communicate between the two parallel avenues to revise the products and to fit to the needs of the market. And so this experience really make me to think about what's really the gap between the laboratory technology and the commercial products. What's the big gap here, and what we can do, how we can get to here to make the good technology to the good commercial products. So probably there are the different paths, and everyone has a different experience. If there's a better options, but the first lesson we have learned really define the right market and the usability. But speaking of the usability, to see the nanosensors, you can see there are quite a lot of applications that can derive from nanosensors. I think the biggest application right now is really the innovative driving force for the agriculture. So it really drives the innovation of agriculture to develop the sensor for the animal plant disease monitoring and the food contamination, the food nutrition, and the refresh needs, and also the water, soil quality as well. And the other important sector is healthcare. So mostly used for the cancer diagnostics and the disease diagnosis and monitoring, and also the biotech development as well. And that also include environment monitoring, as Joe talked about the air quality monitoring. And it can be used for the defense and the security as well. So there are so many type of applications, like really the nanosensors can play the important role there. And but if we want to meet needs from the specific market, and what we can do here. And first, if we think about the food contamination, for example, you want to detect the E. coli from the food contamination, but meanwhile, for the water quality, probably they also need a sensor to monitor the water quality if the E. coli present. For a different purpose, even the same target, the sampling process, the data readout will be different because the metrics, the target metrics background are different. So that really requires the nanosensor to be much more adaptive and can produce the adaptive nanosensors in the different alterations and alternatives very quickly. So how we can produce a nanosensor very quickly with different alterations and alternatives. So that's really the big challenge. And thinking about the economic evolution, so we look at back maybe the 20 years ago or 100 years ago, start from the industrial age, and it really it's for the business efficiency, and then we want to do very quickly, the fastly, in the mass production. And later on, once the internet came out, and it really changed the way of people to gather the data and information. So it's really the information era. And later on, we have more communication apps and the strategy technologies. It really changed the model to the connection economy. So that's really the relationship. And there's more close communication between the consumers, the product manufacturer, to the product developer. So, and if we need to catch this economic evolution by changing the manufacturing process, so how we can manufacture the nanosensors really catch the economic evolution, this is also the big question we can think about as well. And thinking about right now the new technology, the big data, the artificial intelligence, the biomimetics, the 3D printers, cyber manufacturing. So all those type of innovative technology actually can be leveraged to the nanosensor manufacturing as well. So this is really we can think about how the manufacturing evolution that can catch economic evolution. And so here, and compose this figure and to really understand the nanosensor, that's the core material. The nanomaterial, the core part you can use to sense, to detect the events. So here I'm mostly focused on the biological sensor, so to sense the biological events. And there are several measures we care about. That's the specificity and the sensitivity and the detection multiplexing, and provide the better performance of the sensing. And meanwhile, this sensing needs to integrate with the sampling process or target you're going to target, and the readouts, and also the integration to which towards to the usability of the sensor. It's more usable and then more easy to identify particular market. For the dimension of the manufacturability, we need the chemistry and the synthesis, the surface chemistry, and the fabrication, the physical fabrication as well, the nanostructures, and also the engineering to integrate all those together. But adding the other dimension here, and it actually can leverage by the innovative technologies to form this interactive ecosystem for the nanosensor manufacturing. So here, we think about the adaptability because we need to meet the diverse needs from the consumers, from the markets, and we need to produce the different alternatives very quickly. So that requires a production with a strong adaptability. So if we can integrate more innovative technology like 3D printing, which is the additive manufacturing, and also the big data, which help you to build a database and have the machine learning to make the smart manufacturing, and the virtual reality help to have the remote training for the manufacturing process, and which can increase the adaptability of the production and to have some add-on functionality of the end user engagement. And meanwhile, you can have the factory built in the point of use. So in that case, I'm going to focus on the 3D printing here, as we have done some 3D printing work, like help to integrate and package in the nanomaterial to facilitate the nanosensing process. So the 3D printing is the additive manufacturing, also called 3D printing, and we hope they can add another dimension of the end user engaged production. So in that case, we can take advantages and offered by this 3D printing for transforming the nanosensor products. And if there's no radical alterations in the products, for example, it's hard to build the alterations very quickly, but may improve the value delivery for the current products through the additive manufacturing. And also, we can take advantages offered by additive manufacturing to achieve the new levels of the innovation in the products, or even pursue new business models for the end user engaged production. So in that case, you can directly talk to the end user. So that gives you much faster way to improve the product and to meet the needs. So here, there are two examples. I'm going to show here, that's the work we have done before. The one we developed the nanoparticles, and this magnetic nanoparticles integrate and enables sensing of the extracellular vesicles. And then the other technique, actually, we use the 3D printing to print the chips, the device, and integrate with the cell phone, and to print the phone parts as well, to have the smartphone-based sensing technology. So here, the 3D printing really can give you the capability to print the devices and the parts, and even if there's a database available for the end user, and they can access to manipulate or print parts that they need. So we characterize the 3D printing characterization, and there are different type of devices and the microstructures we can print out. And for example, several micro mixers, they are outer mixers without external power required, just based on the capillary force driven to mix the reagents with the nanoparticles. And also, this is the microstructure. We characterize the performance of the printing. So basically, the printing resolution is about 30 micron, so which can give the good structure in the micro scales. And then meanwhile, you can print out the mold to replicate this microfluidic device based on the polymer replication. You can print out the sampling sets as well, that can fit to your nanosensor device, and to have the better sampling. And even there's some innovative way to have the 3D printing the Lego parts, which can combine together to form the Lego fluidic driven system, so which really give you more flexibility for you to play with, and that fit you the purpose. And here I want to show you the why we need 3D microstructure. And the 3D microstructure principally improves the sensing performance. So the first, we talk about sensing, especially for the biosensor. We need to immobilize certain recognition parts on the surface, and which requires the fast mass transfer, and to improve the sensing specificity and the sensitivity. So this is just the plenary structure. It's two-dimensional way. Basically, you won't be able to see the mixing because the laminar flow, and it's diffusion-based. But if you just make a 3D structure, 10 to 90 degree here, and then the mixing is much improved in the same dimension and structure. And then meanwhile, we can integrate nanoparticles, and those nanoparticles integrate into the microfluidic channel, which can be manipulated to give the much faster mass transfer, and to improve the sensing specificity and sensitivity. And we also can improve this fabrication parts to make the production in the point of use. You can print the different parts, and to make the cell phone, the detection system. The first application we used for the smartphone diagnosis of anemia. So you basically can produce those sensors complete in the point of use, and to detect anemia and connect with the hospital. And the other application, we integrate 3D printing device with the nanoparticles here. We develop the surface of nanoparticle with the graphene oxidized polydelphimine coatings, which increase the surface area, and which you can have much better capture efficiency. So we used to capture the extracellular, a lot of nanovesicles. So those nanovesicles also called exosomes, which are very important cargoes for the cell signaling, the biomarker discovery, and the liquid biopsy for the cancer. So we can capture those nanovesicles on the surface of the beads and integrate in the one 3D printing device. And meanwhile, with this smartphone detection, and this smartphone detection can give the two-channel fluorescence color readouts, and you can have this detection and just through the smartphone camera. And based on the antibody affinity banding, and you can recognize the specific marker. And this nanoparticle actually gives the much better detection sensitivity here. And so here there's the examples and show that how the 3D printing, the additive manufacturing can help you to integrate the package, the nanomaterial into the one system combined with the smartphone. And so this gives you much more flexibility and that help you to build usability products in the point of use. So at the end, I know it's right now time, so I just want to give a conclusion and look into the future and hope this can stimulate the discussion and some ideas for us to discuss to find a better path for the production. Okay, so at the end, I want to give the acknowledgement for the funding grant, also my students as well. All right.
Thank you very much, May.